Thank you very much for inviting me for this talk. It's an honor to be here. And as it literally takes a whole crew to work in the deep sea, I would like to immediately thank my collaborator from the University of Brest and Ifremer, where I did the present work. The United Nations proclaimed this decade the decade of ocean science for sustainable development. Martin talked about it in details. And I think we are quite aware today that the oceans hold the key for our climate and oxygen production. They contain 97% of the Earth's water, and they cover more than 70% of the planet's surface. But what we are probably less aware of is that when we talk about the oceans, we actually talk about the deep ocean, and that's the one that holds the key. Because the deep oceans covers 66% of the Earth's surface, and it represents 93% of the biosphere in volume, 93%. So it's absolutely central in ecosystem services, such as sequestration of carbon and heat, for example, crucial in climate change scenarios, or food and energy resources that today mostly happen in the deep sea. And the deep sea is one of our major ecological frontiers. It refers to water below 200-meter depth, where light is insufficient for net photosynthetic production to occur. So there is no algae down there. And besides darkness and limited food resources, the temperature is also pretty cold and the pressure is high. So let's clarify that. At water surface, the pressure is 1 bar. And when one goes down the water column, every 10 meters, one adds another bar. To give you an idea, here in Paris, we are at an altitude of 35 meters. Compared to water surface level, this doesn't change anything for us. The pressure is still 1 bar. But in the water column, in the deep sea, at 35-meter depth, the pressure is already 3.5 bars, and that's already more than 3 times higher. So this just underlines how different marine life is from terrestrial life. And we don't know much about it, because all of these constraints, the deep sea has long been considered as too harsh for life. But scientists discovered relatively recently that actually the deep sea, it's not the deep sea. It's a mosaic of ecosystems, and that we now have ample evidence that there is complex life down there, even abundant sometimes, but based on a completely different biology. And we are pretty ignorant of how it works. And to give you an idea, we currently have high-resolution maps of the whole moon, and the resolution is currently going from 7 meters down to 5 meters. So it means that on the map, you have a dot or a pixel every 5 meters. For the deep sea floor, currently barely 20% have been mapped, and we can see it here in the dark blue. And this gap is only expected to be filled by 2030. And by 2030, the resolution will be to have 1 dot or 1 pixel every 400 meters. So the deep sea represents 93% of our biosphere in volume, and 93% of our biosphere is not understood biologically. And despite our great ignorance, these ecosystems are already threatened by human activities. The resources that lie down there make it a new El Dorado. And one of the major threats is currently impending mining development that will generate light pollution, noise, plumes, and up to habitat destruction. So we know from terrestrial ecosystems that biological rhythms are essential for organisms to survive and for the maintenance of life itself. We also know that there are some key ecological species that are essential also for the ecological balance. But honestly, for the deep sea, we don't know much. We just know that we can disturb or destroy it. So what impacts are human activities likely going to have on the temporal functioning of those ecosystems? And if we impact them, are there some critical thresholds that are key for protecting the animals and those ecosystems? For example, if we talk about colonization or recolonization, what are the critical mass and distances that animals need and the timing of that? To be clear, how many larvae will have to settle at a certain time point to be successful? Knowing that all these processes are highly timed in biology, we know only a few % of the species present in the deep sea, plus there are a lot of cryptic ones. We don't know where they come from. We don't know how they spread. We don't know their physiology. We don't know how long they live. So we don't have the fundamental knowledge to protect them, let alone to restore them. And this fundamental knowledge is today absolutely crucial to meet environmental sustainability in this unexplored ecosystem. And as I said, the biology is highly timed, and that is very important. For example, in humans, rhythm disruption has been associated with diseases like diabetes or cancer. So the temporal variation of organisms condition how we impact them and their capacity to overcome or to adapt to these impacts. So these come with major questions. The first one, okay, organisms are highly tied to their environment. How do they perceive it? Second, how do they tell the time? We know from terrestrial species and coastal species that they possess internal molecular clocks. Are there biological clocks in the deep sea? And if yes, how does this clock function near the limits of life? And finally, how does the temporal functioning of one organism influence the temporal functioning of the organism around it? We know again from coastal or shallow water species that sometimes the rhythm can be relatively weak at the individual level, but very strong at the community level. So again, are there critical thresholds for that? What is the temporal functioning of the deep sea? And we started to have some answer in the last years coming from hydrothermal vents. Hydrothermal vents form in tectonically and volcanically active areas when seawater permeates into the seafloor, goes through the underlying rock and pours out of a vent into the ocean. And there is a whole gradient between the vent fluid and the surrounding seawater where biomass hotspots can develop, and there the life is based on chemosynthesis as a source of energy. And hydrothermal vents are totally blind to the light-dark cycle, but evidence pointed towards, in the last 10 years, tidal cycles at vent in abiotic factors such as current and temperature, or biotic factors such as shell growth or population densities. But the question that I mentioned earlier were not really answered because those evidences were obtained either under artificial conditions or with indirect measures. And one of the reasons of our great ignorance about the deep sea is that it's highly challenging to explore. So I'm a marine biologist, and for today I would like to ask you to dive with me and this remotely operated vehicle to the depths of our planet. And the very first thing that we will see is this. But for today, I'd like to turn the light on a very important species that is the hydrothermal mussel Bathymoduolus azoricus. So this mussel lives on the mid-Atlantic ridge, which is here in the middle of the Atlantic Ocean. And it's what ecologists call an engineered species, because it can represent up to 90% of the biomass on a vent edifice, and it really structures the community. It provides protection, habitat, resources, food. But to start, I needed preliminary data. So we analyzed video recordings over, here gathered over 1 month, from instruments deposited in the EBSO-ABSOS observatory. But even video imaging is challenging in the deep sea. So this acquired 2 minutes video every 6 hours. And despite this limitation, we could show that indeed there are cycles in the deep sea. But that was still a rather indirect answer. So we wanted to take a different approach. We wanted to look at the molecular level, because if you want to understand biological rhythms, one way to do this directly is to look at how the expression of genes is changing over time in the cells. That can be a direct readout, and that was absolutely not easy to do. So first, the deep sea is deep. So you need a boat to go in the middle of the Atlantic, and you need a submersible to dive to the seafloor. Second, depressurization. It's either stressful or lethal for deep sea specimen. And it takes almost 2 hours to bring the sample back on board. And you can only do that once or twice in 24 hours. So this was an absolute no-go for me. I had to look at molecules that could be changing over time. So I really needed to freeze the time of the animals on the seafloor and preserve them directly on the seafloor. So sampling was not easy at all. It was not like collecting mussel at the beach. We had to grab mussels with a robot, to open boxes submitted to 170 times the atmospheric pressure, and to store those mussels into a preservative solution for genomic processing. 2 more constraints. Usually, biologists work under white light because the deep sea is dark. But I needed no interferences, so I needed to work under red light, and that drastically limited the visibility. And last but not least, I had to work over almost 25 hours under a very tight schedule, and that is a real technical feat down there. So the very first accomplishment of my colleague and I was to establish and achieve this very innovative protocol on the deep sea floor. And it's because we achieved that protocol that we could show that, yes, there are biological rhythms in the deep sea at 1,700-meter depth. And to clarify how we as biologists look at it, we sequenced the transcriptome of the gills. So the transcriptome, it's a snapshot of all the genes that are expressed at time t. And we choose the gills because that organ is absolutely key in nutrition and respiration for marine bivalves, and it's a major sensor of the environment. And a common way to look at this, a visual explanation, is this type of figure. So on the x-axis, you have the time from 0 to almost 25 hours. And on the y-axis, each line represents one rhythmic gene. When the gene is highly expressed, the color is yellow. When the gene is poorly expressed, the color is blue. And what we can see on this figure is that in the deep sea, most genes show 2 yellow lines per 24 hours, which means that they are mostly tidal. And tides were also recorded in temperature and pressure data obtained from the vent. We also see that we have a bunch of genes here with only 1 yellow line per day, which means that they show a daily pattern. But like I said, the deep sea is pitch black, and most people walking there use white lights, and they assume that light has no effect on the organism. And I really wanted to challenge that white light assumption because it was so tricky to work under red light. So, importantly, what I just showed you, it's changing when I exposed animals in the lab to light conditions. And we see that we now have less genes with these 2 yellow lines per 24 hours, so less genes that show a tidal pattern, and much more genes that show either 1 line per 24 hours, or yes, 2 lines, but with a very different spacing. So there are more daily genes now. So we show that, yes, there are biological rhythms in the deep sea, and that organisms are not blind to light. And we now want to build up on that and understand the mechanism. So we need to work at the molecular level. This calls for cross-disciplinary science. This goes beyond the normal expertise of an ecological lab. And that's why I started to connect with a lab in Vienna that is this kind of expertise. And to give you an idea, we now, as a preliminary setup, managed to have primary cell cultures for those deep sea mussels. And that will open the doors for all the, at least to all the tools that are commonly used in molecular biology. So we are on our way to answer the major question that I mentioned at the beginning by combining in situ measures advanced with the intrinsic property of the cells. And like I said, this is very important. Biological rhythms are a fundamental property of life. And this rhythm perspective could really profoundly change deep sea science because our work shows that the temporal orchestration of physiology is likely a critical feature even in the most extreme life environments such as vents. So I hope that I made you discover some of the wonders that inhabit in the depths of our planets. There are amazing, fascinating, unexplored ecosystems down there that thrive near the limits of life. And fundamental knowledge is now urgently needed so that we can understand those ecosystems and make informed decisions. Like I said, there is a whole crew behind this, and I want to really thank all my colleagues from the boat, from the ROV, from IFREMER and the University of Brest. I would also like to mention that I have a poster session, so if you want more scientific details or any type of discussion, I will be happy to keep talking about this. Thank you very much for your attention.
Thank you. Fantastic talk. Wonderful.